Thank you very much, Chase, for agreeing to give a Build a Cell seminar, and you can get started.
Okay. All right. Well, thanks for the invitation, Kate, and really great to join this group, particularly one devoted to all things, especially cell-free, which is where my interests come in. So today, given a lot of different things going on in my group, I actually want to tell you a story we published very recently in terms of a general focus of my group, where this is work that was all spearheaded by a fantastic former PhD student, Justin Bento, who's now a teaching professor at Villanova. And so to kick things off, as mentioned, I'll actually briefly introduce myself, where I'll start out. So Chase Beisel at the Helmholtz Institute for RNA-based Infection Research in Germany. And before launching in, actually, I wanted to tell a little bit about my own backstory, since it is a pretty unique one. So for those that haven't met me before, but probably have already guessed at this point, I was born and raised in the US, even though I'm coming from Germany. And so I was classically trained as a chemical engineer, where I went to Iowa State University for undergrad, so not too far from the University of Minnesota. But then got really interested in synthetic biology as it was becoming a thing, and ended up joining an RNA engineering group, one of the first at Caltech, so by Christina Smolke. And there got really excited about all of the great things you could do in engineering RNAs, but also horribly frustrated how many of my designs failed when I shoved them into cells. And so that piqued my interest in natural regulatory RNAs and examples, obviously, that nature uses quite plentifully and could be a great starting point for engineering. And so that took me to the NIH Bethesda, Maryland, where I joined a group studying small RNAs in E. coli. And it was during that time that I was trying to figure out what I was going to do at my next step, that I was reviewing the laundry list of newly discovered regulatory RNAs, thinking I would pick one, and that's the one my group would focus on studying and engineering. And of course, the prominent one on the list at that time was CRISPR, something that at the time was part of this quirky RNA-based immune system, but obviously had a ton of potential that was recognized even at that time. And so when I joined the faculty at NC State University in chemical engineering, that was the focus of my group, studying the biology of CRISPR as an immune system, but then translating it into different technologies. And so about the time that I was going through the tenure process, which thankfully went smoothly, I was hit up about a unique opportunity to join a new Helmholtz Institute in Germany focused on RNA-based infection research. And there were enough reasons and interest behind it that I relocated not only myself, but my family to Europe, and have been there ever since. And so what I ended up joining was this Helmholtz Institute, as I mentioned, that focuses entirely on RNA-based infection research. This idea that we're interested in infection biology, but want to look at the role that RNA plays, as well as how we can use RNA either as a drug target or drug or diagnostic tool. And so, at least most recently, I've been one of 9 different PIs within this institute, which is based right in the middle of Germany in this smaller city called Würzburg. Beautiful town if you ever get a chance to visit, or if you're looking for PhD or postdoc opportunities, some really unique opportunities there. So given that backdrop, what I'll actually talk about today is not explicitly CRISPR. And so instead, I want to focus on a grander interest in my group, which is on how to make genome editing of bacteria easier. Now, part of the motivation behind doing this is this incredible wealth of bacteria that can be found in the natural world, everywhere from our gut to the oceans and the environment, and how we can better understand how they function as ways of improving our own health and livelihood, but also being able to utilize the genetic components that they've evolved over billions of years for our own purposes, again, under synthetic biology. And so coming from an infectious disease standpoint, we obviously want to do this to better understand and even engineer pathogens, as well as members of the microbiome. And then, of course, these microbes also play a massive role in industrial biotechnology as well as agriculture. And so given all these areas, it's really important to be able to genetically manipulate them in order to study their fundamental properties and again, be able to engineer them. But as you look at how people have gone about this process, what's become apparent over and over and over again is that there's this tremendous focus on what you could call model bacteria, ones that people just use repeatedly. And the issue is that while these are great models for studying different cellular systems or as a starting point for engineering, they tend to be the exception rather than the rule. And in many regards, if you want to engineer a microbe, let's say for a particular industrial biotechnological process, the microbes that are going to be of the most interest, the most direct relevance to what you're doing, are not the ones that are models. And this creates a bit of a disconnect as you would like to engineer those, but that's not what people use. And so this ends up being the problem that my group has thought about over a decade now, which is how we can get away from using the easiest strains and actually move on to the best strains. And so behind this, you have to think about, okay, well, what does it take to go from a non-model bacterium to something that you can genetically manipulate and control as easily as you can as a coli, best recognized of model bacteria. And so you'd imagine a general pipeline where you go from just being able to culture them in isolation to getting DNA into them efficiently, to then being able to express things off of that DNA, to finally being able to genetically manipulate the chromosomes of the bacteria in order to change their overall properties and behaviors. And now, admittedly, my group has been really, really focused on this last step, which is once you've gotten DNA in, how do you go in and start genetically manipulating the chromosomal DNA in order to change how the microbe behaves? And as an aside, this really has been a major focus of what my group does. So for instance, we've, especially in the realm of CRISPR, I should say. And so we've tried harnessing these CRISPR nucleases for lots of different purposes, particularly in bacteria, as a way to control how they behave, which genes are expressed. So this includes early work on using CRISPR to turn genes on and off. We've developed a number of different genome editing techniques specific to bacteria. And then we've even been playing with some of these more recent technologies, such as these CRISPR transposons that can insert really, really large chunks of DNA in an RNA-guided programmable way. So lots and lots of capabilities that are being introduced into the bacterial world that allows you to go from a non-model bacterium to something like a coli. But of course, while doing this, we ended up running into a very regular frustration as we did things outside of the coli, which was just getting DNA into these stupid microbes. So on one hand, we had all these great technologies for manipulating chromosomal DNA if you could get the DNA in, but we ran into lots of examples where even that getting the DNA in was proving difficult to impossible. And so it was a personal motivation for our group to ask, okay, well, what's getting in the way there and what can we do about it, particularly as engineers? And so that took us into a separate exploration, trying to understand what barriers bacteria pose to that DNA, and of course, how we can overcome those. And I promise you, we are eventually getting to cell-free systems. So bear with me here. So one of the, well, you can imagine there's different barriers there. Everything from the cell wall being super thick and complicated and a thick barrier to anything larger than small molecules. But then there's also barriers once the DNA gets inside the cell. And in fact, bacteria possess a pretty large and growing assortment of known defense systems that they use in order to distinguish foreign DNA from their chromosomal DNA. All of the idea that the DNA coming in probably has ill intent, probably part of a bacterial virus or a plasmid that's actively replicating through a community, and it's much better off if it just gets rid of it. And so of all the different defense systems that bacteria have, there's a really, really, really common one called restriction modification systems. And these, in fact, have been known for many decades to pose the biggest barrier to foreign DNA. And so how these systems work is that they rely on a combination of DNA methylation and then DNA restriction. So methylation comes from these DNA methyltransferases that will introduce methyl groups onto specific nucleotides within certain motifs. Whereas these restriction enzymes, what they do is they probe looking at those exact same sequences, but trying to see if they lack that methylation pattern. And the idea is that if that DNA coming in lacks methylation at those specific points, that tells the restriction enzymes that's actually foreign DNA and we should get rid of it. And so the restriction enzyme comes in and chews it up. Of course, this was a basis for all of the restriction enzymes that, at least when I was a PhD student or a little bit before, were commonly used in the research lab as a way of being able to piece DNA together or be able to stitch DNA together. But again, it's a defense system that these bacteria possess. And so while you might say, okay, fine, that's an issue, but surely some DNA gets in. The issue is that these restriction enzymes are so efficient at their job that you end up seeing massive drops in the transformation efficiency, again, due to the DNA getting chewed up, if the methylation pattern is not correct. So examples where you get over a 10 to the 7th drop in the number of colonies just by having the wrong methylation pattern. And so you can imagine, especially in non-model bacteria, where you're only getting, let's say, 1,000 or even 100 colonies from a typical transformation. If you're 10 to the 7th below that, there is no way tweaking growth conditions or anything else is going to have you get to the point where you can even get that one colony you like. So this represents a kind of a major problem if you're trying to get DNA into the cell, again, an issue that we encountered. On top of that, what those methylation patterns can look like can be pretty complicated and vary quite a bit as you go from one microbe to the next. So for instance, a given bacterium can encode not one, but multiple of these DNA methyltransferases or MTAses. But on top of that, again, the pattern that you observe can vary even between related strains. And so in many regards, anytime you pick up a new strain, you're almost starting from scratch. So big pain in the butt if you think about how to overcome this. On top of that, it's not just one type of DNA methyltransferase. It turns out there's 3 different types that have been defined, each with its own pretty extensive diversity. And so you have to think about, okay, well, what is it going to take to be able to recreate the pattern created by any one of these? A big headache. At the same time, if you can boost transformation sufficiently to start working with a microbe you couldn't before, it's worth the effort to get across this restriction barrier. And so in fact, for many decades now, there have been a number of efforts and developed techniques in order to recreate those methylation patterns. So just a few traditional examples here. One is that you just go in and you express and then purify the individual methyltransferases, and then you use those for an in vitro DNA methylation reaction with the idea that that can create or recreate the methylation pattern. Another common way is you equip a plasmid propagating strain like a coli with these DNA methyltransferases while getting rid of the endogenous ones. And then that way, any plasmid DNA that you extract from that strain is going to have the right pattern. And so while both of these approaches can work well in principle, there's some really, really big restrictions or issues with their use. Anything from being able to apply them can require anywhere from days to weeks to complete, whether you're purifying the individual proteins or you're trying to clone and express all of these in a coli. And even if you do get to that point, oftentimes these methyltransferases can be heavily toxic just by messing with this methylation pattern of the bacterium. And so some just get crossed off the list almost immediately. On top of that, in most practical applications, people are only doing this with 1 or 2 methyltransferases, but many bacteria have many more than this. So you're at best creating a partial recreation of that methylation pattern. And so given all of these issues, including the ones that my group was experiencing, we really asked ourselves, is there a faster, easier, and more scalable option? And given that question, that's what brought us to TXTL. And so I imagine this group is very familiar with it, but just the key highlights as the important notes is, of course, when we talk about TXTL, this is a cell-free system or solution that recapitulates transcription and translation. And so all you have to do is drop in DNA encoding any RNAs or proteins of interest, and then within minutes to hours, you're producing those. And what's great about this, of course, is that you get around a lot of the issues I mentioned with those other approaches, including not having to purify anything aside from the DNA that you're working with. You're not dealing with live cells, and so any cytotoxicity issues are often thrown out. And because you can do this in half a day, it can be much, much more rapid than what you would have having to culture cells. And on top of that, it's easy to go from a well with a few microliters of these reactions to many wells, as well as scaling this up. And so you can have a lot of different reactions going at once. On top of that, you can actually combine multiple DNA constructs in a TXTL reaction and have all of those faithfully expressed. So lots and lots of great reasons why you could use this. And then, of course, there are commercially available sources, particularly when you're like my group and just are having a really hard time being able to generate the mixes on your own, at least to match what you can get commercially. Now, within these cell-free mixes and given these advantages, there have been a lot of examples of how they're applied. Biomanufacturing was one of the original examples, knowing you could express toxic proteins, but you could also express proteins on, I guess, as needed or on demand, even in resource-limited settings. That actually led to the development of diagnostics based on these cell-free mixes that link the presence of some, say, DNA or RNA in the mix to some sort of visible or measurable readout. And then finally, there have been more recent efforts using these as ways of prototyping different genetic constructs, whether they're synthetic constructs, like trying to build like the repressilator, for instance, or other more complicated genetic circuits, or trying to look at how natural systems are regulated, such as, for instance, flagellar biosynthesis. So there was already a lot to point at why this could be an interesting system as a way of going about recreating these methylation patterns. And so what got me really honestly into this area is this really wonderful connection to the University of Minnesota through a really wonderful collaboration with Vincent Nareau's group, I'd imagine many of you are familiar with. And so he and I actually first met when I was recruited to be part of the synthetic biology summer course at Cold Spring Harbor way back in 2016, and he was one of the other instructors. And as we started interacting, we realized that one, he had this amazing cell-free system that could do stuff in a matter of hours that would take my group days or even weeks to perform. But we saw also a particular opportunity applying this to CRISPR. And so just as a brief aside on the main story, working together, we figured out how to use cell-free systems, or TXTL, as a way to rapidly characterize different CRISPR nucleases, guide RNAs, and systems. And so really this was how I had my first start with cell-free systems and really appreciating what they were capable of. And so building on that early work, we've done a ton of work combining TXTL with CRISPR characterization. And in turn, this has allowed us to characterize actually a pretty diverse set of these CRISPR nucleases with thousands of these present in nature. We were able to take a step further and characterize how phages fight back against these CRISPR-Cas systems through the form of these anti-CRISPR proteins. And then we could also use this as a way of revealing new biology of CRISPR, and TXTL allowed us to churn through some of the initial discovery and characterization work that would have taken much longer using more traditional approaches. And so given this really strong experience and success with TXTL, that led me to ask, can we apply TXTL to this problem with DNA transformation? And so by asking that question, that led us to the setup shown here. And so the idea was that if we want to recreate the unique methylation pattern of a given bacterium, a simple way with TXTL is we can express each of those DNA methyltransferases in a cell-free reaction, and there we don't have to care about toxicity, And then with those produced proteins, we could turn around and perform an in vitro methylation reaction just by including the methyl donor SAM. And the output would be a methylated plasmid we could purify out from our cell-free mix, with the idea that because it has the correct methylation pattern, it'll transform much, much more efficiently into our bacterium of interest. So end up with a lot more colonies than we would have otherwise. And so, as with any technique, you need to come up with a flashy name for it. We came up with IMPRINT, which stands for Imitating Methylation Patterns Rapidly in TXTL. I have to say my group has come up with a lot of acronyms over the years, and this is probably one of my favorite ones, but at least easy to remember and appropriate given that we are imprinting methylation patterns on our DNA using TXTL. And so if you now compare this to those other approaches I had highlighted, the advantages become clear very quickly based on what cell-free systems have to offer. So for one, now any purification requirements are gone. We're just dropping in our DNA constructs. Once we have those constructs, we can actually complete the methylation reaction in a single day, much, much better than what was before. And it has the potential to easily scale from just 1 or 2 methyltransferases to many methyltransferases, matching however many they happen to be in that microbe or the next one. And I mentioned that there are, actually I'll go back and reiterate this, there's these different types of DNA methyltransferases, and how they operate is a bit different. Some operate on their own, like type 2 methyltransferases. Other ones have to be in complex, like with type 3 methyltransferases. And then you have crazy ones like type 1 systems that require this extra specificity protein. And so recapitulating those in vitro is really difficult, but it's something we should be able to do in TXTL. So this was the challenge before us. Can we implement IMPRINT, and does this actually boost DNA transformation in hard to transform microbes? And so we first just wanted to show that we could express different DNA methyltransferases and have them methylate provided DNA out of that reaction. And so we actually started with the coli, mostly because it has 2 extremely well characterized DNA methyltransferases called DAM and DCM. So DAM is a DNA adenine methyltransferase, and then DCM is a DNA cytosine methyltransferase. And they would specifically go after specific sequences modifying the A or C in each of those. And so to read out the methylation pattern easily, we ended up applying 2 established restriction enzymes that specifically go after the same methylated site. One key difference is that one of these, DPN1, that you might have dealt with, only cleaves that site if it's undergone methylation. So a pretty unique restriction enzyme. Whereas the other PSPG1 would cleave the unmethylated site, I should say, for DCM. And so those would allow us to look at whether or not each of those sites undergone methylation. And so here's one of the examples where we've taken DNA that's been either has no methylation whatsoever, one in which we extracted it from our MG6055 strain, so both DAM and DCM were active, and then ones where we had expressed either of the methyltransferases individually or in combination in TXTL, and then in all cases would incubate those with our plasmid DNA. And so just starting out, if you just apply these without any restriction enzymes, you get the typical banding pattern for plasmid DNA in this different form. Now, if you then apply DPN1, which again cuts DNA that's been methylated with DAM, you can see here that you see that, well, not with our unmethylated DNA, but it definitely gets cut if you've extracted it from the host. And then we've introduced DAM either individually or in combination with DCM. And of course, if you just use DCM, nothing happens. Now, of course, when you flip it, you get the other expected pattern. So this is, again, the restriction enzyme that cleaves unmethylated sites for DCM. And so if it's unmethylated, it gets cleaved. The host DNA does not get cleaved. And then get cleaves only if we've introduced DAM, but not DCM in any combination. And so this was a nice demonstration that expressing these methyltransferases and then applying this imprint reaction created the expected methylation patterns, at least based on these restriction digests. And so based on that, we wanted to move into something a bit more complicated, but also an opportunity to demonstrate this boost in transformation. And so what we used was this strain of Salmonella, which on one hand is close to a coli and ends up having effectively the same DAM and DCM genes, but it actually has a number of other restriction modification systems that gave us some headaches, or would give some headaches. So the two to point out in particular are these other types of restriction modification systems. So you have this one written here called LT22, which, just to create confusion, this is a type 1 system. And so it actually has a methyltransferase and a specificity protein that have to be expressed together to methylate DNA. And then a type 3 restriction modification system where you have the restriction enzyme and then the methyltransferase that are normally combined. And so what we did is we expressed each of these in TXTL and then not only evaluated methylation through this more restriction approach, but then actually looked at what happens to the number of Salmonella colonies we get. And so this was the larger data set here that I'll walk through. And so we just set our baseline when we don't introduce any methyltransferases, which we set to a value of 1, but I can tell you was a pretty massive drop compared to what we would have if we were transforming a coli. And so what happens now is you add each of these methyltransferases alone, you either get no boost whatsoever for DAM and DCM, but then with our type 1 system, you get the largest boost, about 100-fold increase in transformation, and then a smaller but still substantial increase with our type 3 system of about 5-fold or so. And what was really nice to see is that if we combine these two together, we get an even larger boost in transformation. This idea that we're methylating more sites that these restriction enzymes would try to cleave, which would otherwise reduce the number of transformations. Now, what was interesting is we started tacking on the additional methyltransferases, whether it's DAM or DAM and DCM, we actually started seeing a drop in the transformation. And what we ended up figuring out was that this type 1 system here really has issues when there's other methylation reactions taking place. It's just really slow and then peters out quickly. And so just to summarize one quick point is that a big reason behind this is that these methyltransferases, at least for type 1 systems, prefer to methylate DNA that's hemimethylated, namely that one of your two DNA strands already has that methyl group on there. And so because of that, and because we're giving it naked DNA, it doesn't have that, and it's a less preferred substrate. And so we were able to use mutations already reported in the literature to alleviate that issue. And so as a result, we were able to actually boost the transformation up to, or pretty close to, the levels if we just used post-methylation DNA to begin with. So we saw this as a really nice example where you could not only apply imprint across a pretty complicated set of DNA methyltransferases, but you could actually use this to boost transformation upwards of 1,000-fold over what you had previously, but now to the point that it's approaching what you would get if the DNA had actually been extracted from that bacterium to begin with. Now, for Salmonella, that was manageable, but as you look at other examples, there's lots and lots of other combinations that you would want to test. And depending on the bacterium, transformation alone is a pretty annoying procedure, particularly if you're working with anaerobic bacteria, for instance. And so it'd be really great if we had a more compact, streamlined experiment that allows us to test lots of those different combinations in one pass. And so to Justin's credit, devised a high throughput approach in order to test a lot of those combinations in one experiment. Briefly, the way he did this was he had his shuttle plasmid that could go into the new bacterium, but then could introduce a set of unique barcodes. And the idea is that one or a few barcodes would be matched to a given combination of DNA methyltransferases. And so the idea is that if you transform these in, some of those methylated plasmids will persist much better because they're not being cleaved as much. And you'll end up seeing them much, much more frequently than others, whether you plate cells and extract colonies, or you grow the cells in liquid culture. And so the idea is that just by looking at the change in abundance of those barcodes, we can back out which methyltransferase combinations were good and which ones weren't. And so the place that he started was Bifidobacterium breve. So it's a common Bifidobacterium strain used for probiotic research and development. And that was an area that Justin had a personal passion. He really wanted to get into microbiomes and gut health. Unfortunately, Bifidobacterium is a strictly anaerobic bacterium, not fun to culture, and definitely not fun to transform. And so definitely set him up for a harder challenge. The nice thing about HT-Imprint is it gave him the opportunity to apply this high throughput approach and then get all the information he needed out of one experiment. So in this proof of principle, I should point out too, the cohort of methyltransferases in this bacterium wasn't too bad, but still a decent set. So there were 3 of these total where he would just express the DNA methyltransferase. And the question was, which of these would boost transformation? And so what I'm showing here is the output of HT-Imprint, where darker blue represents greater enrichment of the associated barcodes. And so you can see when he tested either no methylation or individual methyltransferases or combinations thereof, combining all 3 definitely had the greatest benefit. But having 2 out of the 3 was very closely behind. And in fact, if Justin went back, or when he went back and then tried these individually to validate HT-Imprint, had a very similar trend. So namely looking at that boost in transformation using those top 2, so U1 and U3, as well as using all 3, gave virtually the same transformation boost. And in fact, it was the same as if we had extracted DNA from the host, which again, should be our ideal methylation pattern. And in this case too, it resulted in 100 times as many colonies as if we didn't use this approach to begin with. So already making work with Bifidobacterium much, much easier. Then we moved on to a much more complicated one, which is from Bifidobacterium longum in this particular ATCC strain. And so on one hand, you look at this and say, okay, only 2 different restriction modification systems, not too bad. What complicates matters, though, is that one of them has 2 different methyltransferases that are encoded. And then another one of these type 1 systems, has 2 different specificity proteins. So it would direct the methyltransferase to 2 different sequences. And so really, there's actually 4 different restriction modification systems that are present here that we have to manage. And so when Justin applied this approach and trying a whole set of combinations, it turned out the takeaways were much more complicated. I won't go into all of the details, but you can see that the combinations we tried went from being really good to bad, without it being clear why some work better than others. What was nice, though, is at least we could pull out certain ones that seemed to work pretty well as different combinations that we would want to try to reproduce. And so what was nice is that when Justin tried just a small set of these, again, because this bacterium is a pain in the butt to culture and transform, was able to show that just using certain combinations that we had pulled out, we were able to get upwards of 60 or 70-fold increase in the number of colonies out of this. So definitely well worth the effort that Justin put in it. As a kind of a final point to start wrapping this up, of course, Justin went through this process to develop specific methylation patterns for Salmonella, or these 2 different Bifidobacterium strains. But one relevant or reasonable question is, well, if we created a methylation pattern here, would that work just as fine over here or over here? Maybe you just need to create one methylation pattern that ruled them all. Brief Lord of the Rings reference for you. And so that's what we specifically wanted to try. Namely, if we recreated these methylation patterns that were specific to Salmonella and then either of these bifidobacterium strains, what impact do they have on transformation in each of them? And so when you consider all the different combinations, we ended up with something that looks like this. And so in this case, anything in blue means good transformation. White is the same as if we would have transformed unmethylated DNA, and green means it actually got worse. So unmethylated DNA would have been better. And so hopefully from this, you can see that in general, when you run down the diagonal, which is to say we matched up the methylation pattern with the strain, you got what were the best boosts in transformation by following that matchup. But the moment you deviated from it, it either got worse or much, much worse. I think one of the best examples is shown here, where if you took the methylation pattern from one bifidobacterium and transformed it into another, it was actually much, much worse than just using unmethylated DNA. And so I think that the takeaway from this is that, and really an argument for imprint, that you really need to match the methylation pattern to your bacterium of interest rather than expecting to end up with a general pattern you could use broadly. So that's the nature of microbiology, but at least we have a scalable, high throughput way to address it. And so kind of to wrap up, taking these microbes, being able to transform them more efficiently using this cell-free approach actually opens up a lot of really, really nice opportunities in the realm of bacterial genetic manipulation. So I think one important point, well, actually, aside from these, is that a bacterium that was already hard to transform for one reason or another, you're able to boost the transformation, which is really, really awesome if you want to study this microbe versus this other one. At the same time, even when you have that microbe and can transform it, there's benefits of being able to get even more colonies. So for instance, if you wanted to encode larger constructs, such as entire biosynthetic pathways, you're going to end up with really large plasmids that take a huge hit in the transformation efficiency. So this is a way to compensate for that. On top of that, if you want to do genome editing, most approaches involve killing off unedited cells. That's the standard approach with CRISPR. And so you end up getting a massive hit in the number of colonies. And so anything you can do to increase those numbers increases the odds you're going to end up with what you wanted. And then the final point, and something we actually demonstrated in the paper, was that you could use this to transform libraries and end up with many, many, or much, much better coverage of that library than you would have without applying the imprint approach. And that actually makes it possible to do the library screens that you wouldn't have been able to do otherwise. Okay, so hopefully just a few things I've described to you that become possible in applying the cell-free approach. And so hopefully the few things that you'll walk away with, first of all, that if you want to start working with or trying to create a new model bacterium or just work with a given bacterium of interest because it's the best fit for your application, there are some real barriers there to overcome, including how to transform them as well as how to genetically manipulate them. And the hope is that imprint just makes it a lot easier to transform these bacteria as one stepping stone in getting through this pipeline I showed above. And then the final point is hopefully you appreciate that we were really started with just this incredible appreciation for what TXTL is capable of and saw one particular problem where it was suited. And I think the challenge to all of you is that TXTL can be used so much more broadly than this. Particularly in areas that we work, I feel like there are endless examples where we just have the time and bandwidth. We could be applying TXTL to make things easier, better, faster, and so on. And really, the sky's the limit at this stage, how it can be applied in that regard. And so consider this one example that will hopefully inspire you to think of other examples that could be pursued using these systems. All right, so with that, I'll conclude here. I already talked about Justin and his incredible work that made this all possible. Also mentioned this really wonderful collaboration with Vincent Moreau over the years. And of course, this representing merely one of many different things my group is working on. So with that, I will stop here and happy to take any questions.
Thank you so much. I really love this work. There is several questions in chat. Can you see chat?
How about this? Is it okay if I just exit out of the screen? And it'll also be easier to see those. Okay, I'm seeing the chat now.
If you can
All
read
right.
a question out loud, that's going to go for the recording.
Okay, great. All right, so I'll start with the first one and then go down from there. So have you tried making cell-free lysates for the bacteria you're trying to transform? Okay, excellent question. And you can imagine like that's a whole field in itself, how you start with microbe X and then make a cell-free mix out of it. And in fact, in this whole area of trying to recreate methylation patterns, that was one of the easiest things people have tried. You create a lysate and rely on the DNA methyltransferases that are already active there. The problem is that those same microbes also have plenty of active restriction enzymes. And so when you stick your DNA in, the vast majority of it gets chewed up. And then all these other issues come up in trying to purify it out. And so even though that could be a viable approach for this microbe versus that one, it ends up causing a lot of headaches, and why we thought, let's start with just our generalized cell-free mix and then just bring in the DNA methyltransferases. So Mohammed, hopefully, okay, well, that was the first part. Let me see what the second one was. It says, or what are some challenges for making the lysates that made you not take that route? I feel like it would be really useful information for the experiments you've been running to validate pre-transformation if DNA will get degraded or not.
Yeah, I feel like
okay,
you already
right.
answered that question in the first part, so yeah, I don't have to answer that.
Okay, all right, thanks, Mohammed. Yeah, I like the way you were thinking. I was obviously thinking along the exact same lines. All right, so next one is from Carla. All right, thanks for mentioning the awesome work. Did you ever try imprint with eukaryotic methylation patterns for transfection of yeast or mammalian cells? Ooh, the quick answer is no, we have not. Now, admittedly, at least as I understand it, DNA restriction, especially based on methylation patterns, is not an issue in yeast or mammalian cells. They have other ways of recognizing foreign DNA. At the same time, you can imagine applying TXTL as a way to study those methylation patterns and what they mean for those instances in which methylation is important. Or maybe that methylation is important based on how those genes are expressed and regulated. And to my knowledge, I don't think that's ever been tried before. So there could be some really kind of unique opportunities there, even if it's not related to boosting transformation. All right, should I keep plowing ahead, Kate?
Yes, please do.
Okay, all right. Two more on here. So the next one is from Tanner. So could your method work for genome transplantation, like for JCVI, mycoplasma, stem cells? Yeah, great question. So I would say, in short, at first glance, the answer is very yes. If anything, I think this would be a really, really good approach to apply that, because it's all being done in cell-free without having to go through any membranes. And if I remember from a lot of that original work where they had synthesized the entire genomes, methylation was an issue, at least in one or two of the cases. And as you can imagine, doing this as an add-on to make that process even easier. So great example there. Of course, it hasn't been done, hopefully, yet. Last one from Ilyan. So nice work, thank you. When I search the RM systems of some E. coli strains, there are many methyltransferases that recognize same site. Is there any difference in these methyltransferases to methylate sites? Okay, so I'll try to attempt this one. So, well, first of all, as you look across really diverse E. coli strains, including things like environmental isolates or even clinical isolates, they could actually have pretty widely varying sets of restriction modification systems. And while in general, transforming E. coli is thought to be pretty easy, you can run into these very real exceptions where they do have a different complement of restriction modification systems that make them really hard to transform. And so there, I would expect imprint to be useful, but also just representing yet another example where these restriction barriers really get in the way of introducing DNA, even though we would call those E. coli strains. Now, I think maybe what you're referring to as well is that E. coli pretty commonly has DAM and DCM, and so you're going to find that across the board, but they don't seem to be playing any role in DNA restriction. They actually seem to be involved in regulating gene expression across the chromosome, as well as having some role in DNA repair. And so these DNA methyltransferases can serve in other roles or functions beyond restriction, and that's something that can also be sorted out using our method. So hopefully that answered that question.
I think you went through all of them, unless we missed something. No, I don't think we did. Thank you so much. Yeah, I'm actually curious myself about the whole genome transplantations. That's something we're working on now too, and the yields of that are just horrible. If we can do something to boost that, that would be a huge help.
Yeah, well, I can speak to that briefly, especially since it looks like we have just a little bit of time. Well, let me ask you this actually. What microbe or microbes are you going into in this case?
Mycoplasma.
Oh, okay, all right. So I do know that they have their own sets of restriction modification systems. I'm thinking about the right organism. Now, can you introduce plasmids into those bacteria or in them as well?
Reluctantly.
Okay.
They'll do it, but they'll grump about it. That it's not the easiest system to transform.
Fair enough. Well, if anything, it's honestly a great starting place. I mean, admittedly for imprint, it's great if restriction modification is a barrier, but it may not necessarily be a barrier, the biggest barrier you encounter. And so if anything, in applying this approach, one thing I've advocated for from people I've talked to is just trying to figure out, is restriction an issue? And so there are some simple things you can do ahead of time, even before launching into even our cell-free approach, which I do feel is more straightforward. So that'd be the first question. Is restriction an issue? And if it is, then I think imprint could really benefit you.
Mm-hmm. Yeah, I might get back to you on that, because I feel like we should be poking in that direction to get something to work better than it does right now. All right,
Yeah,
last call for
feel
questions.
free to reach out.
Thank you. Any other questions? If not, I want to say thank you so much again for a fascinating talk, and thanks for giving basically a second talk answering questions. And have a great rest of your week.
My pleasure.
Bye, everyone.